एनर्जी करेंसी की बात करें सेल की, तो एटीपी इज रेफर्ड एज एनर्जी करेंसी ऑफ द सेल। आज हम इसकी बात करेंगे स्ट्रक्चर के पॉइंट ऑफ व्यू से, एंड देन फंक्शन जो है, देन वी विल कंटिन्यू विद द फंक्शन। Before going into detail of structure of the mitochondria, we somehow talk about aerobic to anaerobic to aerobic mode of respiration, toxicity of oxygen की अभी थोड़ी देर पहले बात की, उसको कंटिन्यू करते हैं। आपने एवोल्यूशन में पढ़ा होगा दैट फॉर क्वाइट अ लॉन्ग टाइम हाइड्रोजन वाज द रेस्पिरेटरी गैस, and that hydrogen, those organisms at that time, they were using hydrogen as a gas for metabolism। और उस वक्त जो है, अगर हम टाइम लाइन के ऊपर बात करें, तो इट वाज अबाउट 2 billion years ago, दिस वाज द सिचुएशन दैट हाइड्रोजन वाज कंसिडर्ड एज द gas, molecular hydrogen for purpose of metabolism। आज भी अगर देखा जाए तो ऐसी सिचुएशन एग्जिस्ट करती है जिसको आप एनारोबिक रेस्पिरेशन कहते हैं। अगर मैं ग्लूकोज मेटाबॉलिज्म की ही बात करूं, ग्लाइकोलिसिस इज बेसिकली एन एग्जांपल ऑफ एनारोबिक कंपोनेंट ऑफ एनारोबिक मेटाबॉलिज्म। और अगर आप ग्लूकोज का एंड प्रोडक्ट देखें तो दैट इज पायरुवेट। और ग्लूकोज में एनर्जी प्रोडक्शन इज वेरी माइनर, बट मोस्ट ऑफ द एनर्जी इज स्टिल प्रेजेंट इन पायरुवेट विच कैन बी मेटाबलाइज्ड इफ प्रोवाइडेड विद ऑक्सीजन। और वो जो है मेटाबॉलिज्म कैसे होता है, देन फॉर दैट पर्पस द पायरुवेट हैज टू गो टू TCA or Krebs cycle, and from Krebs cycle the products of the metabolism, they have to go to electron transport chain in the mitochondrial membrane. So then at the end of electron transport chain, we come up with water and energy ATP. And water is the simplest compound, simplest product which don't have any more stored energy. So this is the overall picture जो कि इस पूरे चैप्टर में हमारे बार बार बात करेंगे। So about 2 billion years ago, hydrogen was the gas, respiratory gas. Then there appeared cyanobacteria, which were like using molecular oxygen, sorry, which were using water molecule and split the water molecule to produce the molecular oxygen. So in other words, we can say that about 2.4 to 2.7 billion years ago, there came another creature who was responsible to release extensive quantity of oxygen into the atmosphere. And this was the beginning, or this was this, setting the stage for the shifting of anaerobic to aerobic mode of respiration, because with the passage of time, the oxygen concentration, oxygen in the atmosphere started getting increased, and then this oxygen gradually replaced hydrogen as the respiratory gas, because with the passage of time, these organisms, they shifted their respiration to oxygen, so that there was development of further metabolic pathways, which made it possible that the stored energy at the end of the anaerobic pathway can still be retrieved by the metabolic processes in the cells. One of the example, if we talk here, is glucose to pyruvate, pyruvate to pyruvate to NADH or FADH2 through TCA cycle, and then electron transport chain के जरिए at the end of the water. These two equations, these are important. अभी हमने बात की है कि molecular oxygen जो है is toxic. Molecular oxygen toxic क्यों है? Because this is ready to take electron from any macromolecule present in the cell, and it becomes highly reactive. शायद आपने किसी जगह पे reactive oxygen species के बारे में पढ़ा हो. If these molecular oxygen is present in the cell, it can destroy the cell, even it can kill the cell. So this is the pathway how it happens, that molecular oxygen binds with the protons, producing the hydrogen peroxide. अब यहाँ पे हाइड्रोजन peroxide का जो है वो डिटॉक्सिफिकेशन जो है वो पेरोक्सिसोम में जाके होती है. So when we talk talk about evolution of the membranous organelles, so all the membranous organelles have their functions related with each other. अगर एक जगह पे कोई product बन रहा है, that may be the starting material for the functioning for the functioning of the second organelle. So toxicity of oxygen के हवाले से मैं पहले भी बात कर चुका. Then we proceed further with the structure and function of mitochondria. If we look at this picture, we can see an electron micrograph of the mitochondria, which is present within the eukaryotic cell. The size is so much that we can see it with the light microscope, and depending upon the cell type, the mitochondria may have different structure, but their primary function will remain the same. That is the energy production or powerhouse of the cell. If we look at the size of mitochondria, it ranges from 1 to 4 micron meter in length. These mitochondria. और ये एक स्पेक्ट्रम का एक एंड है दैट इंडिविजुअल माइटोकॉन्ड्रिया दे हैव 1 to 4 micron meter length, but the other side of the spectrum is that the mitochondria has the ability of fusion and fission. शायद आपने ये चीज़ पहले ना सुनी हो. That indicates that mitochondria can come and they can fuse with one another in such a way that you can see the branching network of the mitochondria. You can see this blue picture, and 0 second के ऊपर देखिए, this blue line represents the mitochondria which are fused together to make up quite a long mitochondrial branching network. After 3 seconds, you can see there is some change in the shape, and as the time increases, you can see there is dynamic change in the arrangement of the mitochondria. So mitochondria has the ability to fuse as well as it has the ability to fission, means they can separate from each other, giving sometime an appearance of branching pattern. Then about the question of fission, that how it is possible that mitochondria can split into two structures. For that purpose, if you look at this picture, this green stuff is basically the endoplasmic reticulum, and the tubules of endoplasmic reticulum, they are mainly responsible to come up and to make a loop around these elongated mitochondria. Once this structure is at its position, then there comes some proteins which are DRP1 proteins. They take their position at the fission site, and you can see that they perform the contraction function, ultimately breaking down the mitochondria into two components. Keeping in mind the function of the cell, the mitochondria has special arrangement where the most of the energy is required. If I talk about the paramecium, and you can recall that the paramecium is a ciliary microorganism, have large number of cilia present over the body, which beat for the movement. The energy required for this mobility is derived from these mitochondria, and for that purpose, mitochondria are arranged below the plasma membrane in that particular unicellular organism. Similarly, if we look at the this picture, this is the head of this is the tail of the gamete, male gamete, and you can see these black dots. Basically, these are the arranged mitochondria, and they are the source of energy for required by the tail of this gamete. Besides these function of energy production, mitochondria also play a certain role in different function. For example, maybe you have studied about sarcoplasmic reticulum. Sarcoplasmic reticulum is a storage house of calcium. हमने इसके ऊपर बात की थी जब एंडोप्लाज्मिक रेटिकुलम की बात कर रहे थे. Besides endoplasmic reticulum, this organelle is also involved in storage of calcium for the cell. And then there are some other functions, probably, yes, some thing related with the, oh yes, these are the sites for oxidative phosphorylation for the supply of ATP in the plant cell as well during dark. Plant cells may you might have had idea that there occurs photophosphorylation in the plastids where the ATPs are produced. This is the major difference between plastids एंड माइटोकॉन्ड्रिया के प्लास्टिड्स में जो है वो फोटोफॉस्फोरिलेशन इज अ प्रोसेस. यहाँ पे जो है वो ऑक्सीडेटिव फॉस्फोरिलेशन इज अ प्रोसेस. आगे चल के मैं इन दोनों के डिफरेंस की बात करूंगा. That they are the source of ATP. But what happens when there is no light available to the plants? Then from where the energy or ATP comes? Then they at that time the energy or ATPs are provided by the mitochondria. Mitochondrial membrane. This picture shows you the structure of the mitochondria, the outer membrane, the matrix, and these foldings, which are known as cristae. I will go a little bit in more detail about these structures. The outer membrane and the inner membrane, they are separate. They are different from each other, uh, although they are having more or less similar composition, lipid and protein composition, but the percentage of lipid and proteins, they are different. Then presence of certain specific channels, integral proteins, they are different. The inner mitochondrial membrane can also be divided into two zones or into two domains. One domain is called as inner boundary membrane. This membrane, which lies below the outer membrane, is called as inner boundary membrane because it makes up the boundary for this mitochondrial matrix. Then if we look at the inner boundary membrane, it is rich with the proteins which are responsible for imports for the mitochondrial proteins. Because you might have an idea about the mitochondrial function, that how many type of enzymes it may require for Krebs cycle, for beta oxidation cycle, for electron transport chain, and for a number of other functions. Then the other domain is, or the second part of this inner mitochondrial membrane is this invagination, जिसको हम क्रिस्टी के नाम से जानते हैं. And these cristae are linked with the inner boundary membrane by a cristae junction. So पहले आप लोगों ने इसको इस तरह से पढ़ा होगा that inner mitochondrial membrane has invaginations in the and makes up the foldings within the mitochondrial matrix, which are called as cristae. Yes, this is somehow the same, but the junction is important over here as well. So we divide the inner mitochondrial membrane into the inner boundary membrane and then uh, inner foldings, which are called as cristae, and we say that these cristae are linked with the inner boundary membrane through cristae junction. Besides this, the energy transducing systems, that is the cytochromes और इलेक्ट्रॉन ट्रांसपोर्ट चेन के जो complexes हैं, they are associated with these cristae. And with these cristae, there are attached the F1 particle, which are the sites for oxidative phosphorylation. इनको यहाँ पे हम ATP synthase particles भी कह सकते हैं, F1 particle को, because F1 particle is responsible for the production of ATP molecules. That will also be discussed later on, that how the ADP is converted into ATP. Then जो membranes हैं, इनर बाउंड्री मेम्ब्रेन जो है, अगर हम इन मेम्ब्रेन्स को के फर्दर रोल की बात करें, जो इनर मेम्ब्रेन है दैट डिवाइड्स और दैट प्रोवाइड्स द two different segments or two different compartments within the mitochondria. One compartment is this one, which is called as matrix, and the second compartment, which is present in between the outer mitochondrial membrane and inner mitochondrial membrane. This is referred as peripheral space. This peripheral space has very significant importance because it will play its major role during oxidative phosphorylation. आगे चल के डिटेल में इसकी बात आएगी। ये अभी तक के लिए कोई क्वेश्चन है तो पूछ लीजिए। Okay. Then we compare the outer mitochondrial membrane and the inner mitochondrial membrane, because outer mitochondrial membrane is the place of entry for anything from the cytosol, and inner mitochondrial membrane is responsible for the functioning of the mitochondria. If we look at outer mitochondrial membrane, 50% lipids by weight are present in outer mitochondrial membrane, और इसके अलावा जो है certain enzymes are present for different activities like oxidation of epinephrine, degradation of tryptophan, or elongation of the fatty acid. These are the functions which are performed by outer mitochondrial membrane. Outer mitochondrial membrane can be related with the bacterial cell walls, because we believe that these mitochondria were once bacterial cells, which during the course of evolution became the part of evolving eukaryotic cell. So when we compare this chemical composition of outer mitochondrial membrane, this is homologous with the bacterial cell wall, and this membrane has certain specific proteins which are referred as porins. If you look at this picture, this is a protein which is a porin protein. Why these are called as porins? Because they are pore like proteins, and they are quite much they are not static structure, rather they are dynamic structure, because mitochondria can control the opening and closing of these porin proteins, so that they can allow the entry of different biomolecules into the mitochondria. For example, अगर हम बात करें NAD key, because NAD is required for oxidation reduction reactions in the Krebs cycle. Then coenzyme A to convert the to produce the acetyl coenzyme A, and then at certain points we may need ATP for as a source of energy. So all these biomolecules, they enter from these porin-like proteins into the mitochondria. जब हम बात करते हैं इनर माइटोकॉन्ड्रियल मेम्ब्रेन की, देयर आर अराउंड 100 different polypeptides present in inner mitochondrial membrane. Keep in mind the function performed by inner mitochondrial membrane. There are so many cristae, there are so many electron transport chains. Each electron transport chain has many cytochromes. So the protein component or protein percentage will be higher in the inner mitochondrial membrane as compared to the outer mitochondrial membrane. और अगर हम नंबर वाइज देखें, so for every single protein we will have one protein for every 15 phospholipids, which is quite much high number of proteins as compared to any other membrane. There is no cholesterol present in the inner mitochondrial membrane, and large quantities of phospholipid, that is cardiolipin, is present in the inner mitochondrial membrane. And inner mitochondrial membrane can be compared with the plasma membrane of the bacterial cell. So two layers here in mitochondria, two layers in the bacterial cell, they are related with each other. जब कार्डियोलाइपिन के हवाले से बात की जाती है, तो इसका जो फंक्शन है, it is to facilitate the activity of several large protein complex, which also involve the electron transport and ATP synthesis. ये हमारे functional aspect में जब हम बात करेंगे वहाँ पे clear होगा। और इसके अलावा जब हम permeability के हवाले से बात करते हैं, the inner mitochondrial membrane is permeable for all the molecules and ions. We need specific specific channels, specific transporters, so that the ions can cross the inner mitochondrial membrane. We will talk about it when we will talk about the oxidative phosphorylation. Then comes up the matrix. Mitochondrial matrix may mainly the enzymes are present. Besides the enzymes, there are present many molecules of DNA, circular DNA, just like a bacterial cell. There is present circular DNA in bacterial cell. The same circular DNA or similar kind of circular DNA are present in the mitochondria. It has their own RNA and protein synthetic machinery. हमने राइबोसोम्स माइटोकॉन्ड्रियल राइबोसोम्स के हवाले से ऑलरेडी बात कर ली हुई है. जो proteins हैं, not all the proteins required by the mitochondria are translated within the mitochondria. In human, there are 13 proteins which are translated by the mitochondrial DNA, whereas rest of the proteins they are encoded in the cytoplasm, and then they enter into the mitochondria. Besides this, human mitochondrial DNA encodes for two ribosomal RNAs, which are part of the mitochondrial ribosomes, and 22 transfer RNA molecules, which will be used for translation within the mitochondria. Mitochondrial DNA जो है, that has significant importance in the history because of its evolution or evolutionary relation with the bacterial cell. With the passage of time, अगर हम ancient bacterial cell की बात करें, most of the proteins जो bacterial cell खुद जो है translate करते थे, with the passage of time, those proteins were lost by mitochondria itself, and mitochondria became dependent on the genomic DNA, nuclear DNA for those proteins, and only those proteins are now translated by mitochondria, which are hydrophobic, and they are not they cannot enter the mitochondrial membrane if they are allowed to be synthesized in the cytoplasm. So, in other words, we can say that all the hydrophilic proteins they are translated in the cytoplasm by the nuclear genes, and all the hydrophobic proteins they are translated within the mitochondria. जब हम पॉलीमरेज का कंपैरिजन करते हैं, हमने आरएनए पॉलीमरेज जो है प्रोकैरियोट्स में भी पढ़ा. एक पॉलीमरेज मल्टी सब यूनिट होता है. हमने यूकैरियोट्स में तीन किस्म की जो है वो पॉलीमरेजेस पढ़े थे. दिस आरएनए पॉलीमरेज इज नॉट सिमिलर विद एनी ऑफ दीज टू. ना तो प्रोकैरियोट से मिलता जुलता है, ना ही यूकैरियोट से मिलता जुलता है. रादर दिस इज मोर सिमिलर विद वन ऑफ द bacteriophages, which is the bacterial viruses. Now, what is the evolutionary significance is still a question. So we can simply say that the RNA polymerase is more closely related with the bacterial virus, which is a bacteriophage. The last point about the mitochondrial DNA or the structure, that mitochondrial DNA is quite frequently used for ancestry determination. People normally use this DNA, sequence it, and correlate it with the different tribes, with different regional populations, to compare the evolution of different populations from around the globe. One of the example I can quote here, that the gypsies, which live in Pakistan, they are related with the red Indians of United States. There has been a report on the DNA studies that the gypsies which are living in the subcontinent in Pakistani regions, when you trace back their lineage, they are related with the red Indians of the American continent because of similarities in their genome. If you want to trace your origin, there is possibility that different companies they can take your mitochondrial DNA and they can run it to determine that which lineage your mitochondrial DNA is more closely related.